Good morning, good afternoon, good evening to those joining us, and welcome to the BioCelerate Collaboration Forum. And for joining today's discussion surrounding the use of SEND. Today's webinar is entitled In Comparable Terms, Driving Efficiencies to Simplify the Ability to Detect Class Effects in an Automated Manner Through the Use of SEND. We could go to the next slide. Today's agenda will include a brief welcome from myself, including logistics and ground rules, and then we will dive into the topic at hand regarding cross-study analysis in terms of our approach and our analysis and the results and why this is important to the industry. Next slide. For today's webinar, all participants are muted for this call. For your audio, please either feel free to use your phone or your computer. To answer or to submit a question, please use the question tab up at the top right-hand part of the screen there. We will hold all questions until the end of the presentation, and then we will address your questions at the end. Also, we have a link at the upper right-hand corner, as you see there, including the handout. And in this handout, you will have links to all the relevant materials that we will be speaking about today as well. And this is a reminder that the webinar may be recorded in whole or in part. And if you do not consent to that, then we ask that you please feel free to leave the webinar now. Next slide. In terms of the ground rules, a few basic points that we want to remind all of our participants on. Today, the topic at hand revolving around SEND and cross-study analysis, participation in that is in implementing that is completely voluntary, as is using all BioCelerate assets and tools. There are several things that we cannot discuss here today, and they are listed there for your reference. And likewise, we cannot answer any questions about those particular topics as well. Next slide. And just to briefly set context, for those that aren't aware, BioCelerate is a not-for-profit entity created to foster collaboration focused on the clinical space within the pharmaceutical industry. BioCelerate is a subsidiary of BioCelerate, and that was created a few years after the inception of BioCelerate to address and create collaboration in the non-clinical space. So this presentation is being brought to you by BioCelerate, but obviously we welcome anybody from any part of the development value chain to participate here today. Next slide, please. This is just as an acknowledgement for all the participants that joined in the BioCelerate and FDA collaboration regarding the SEND harmonization work that you're going to hear about today. This has been a long journey culminating in this final endeavor, and it actually has moved a lot of the efforts forward in terms of greater SEND utilization beyond just the standard itself. So congratulations and thank you to all of these team members. Their dedication and hard work has certainly helped to advance that objective, and we thank them. Next slide, please. And now it is my distinct pleasure to introduce our panelists for today's collaboration forum. I'll start with Mark Carfagna. He is a toxicologist at Eli Lilly and Company, where he designs and executes safety assessment strategies for compounds in all stages of development. Over the past 10 years, Mark has also been involved in efforts to use SEND to enhance study interpretation. He has led SEND initiatives for both BioCelerate and Fuse, and collaborated with CDISC to create data use standards. And Mark has been with BioCelerate since its inception 8 years ago, approximately. Bill Hauser works as a senior principal scientist at Bristol Myers Squibb, where he leads the System Administration and Data Analytics Group for non-clinical toxicology. During roughly half of his 34 years at BMS supporting toxicology, he has held pivotal roles in the development and use of the SEN standard, both internally and through industry consortia such as BioCelerate, Fuse, and CDISC. And lastly, we welcome Stephanie Lewin-Roth-Quinn, who earned her PhD in pathobiology from Brown University and then continued her scientific career focusing on human health and disease research with direct experience in the development of anti-inflammatory and chemotherapeutic small molecule therapeutics at Yale University. Stephanie joined the FDA in 2009 within the former division of metabolism and endocrinology products as a pharmacology toxicology primary drug reviewer and is currently an associate director in the Office of New Drugs Immediate Office. She is involved in numerous projects related to pharmacology and toxicology policy, process, and modernization efforts, including the use of the SEN standard. So with that, I will turn over the presentation to Stephanie to take us through the first part of the topic. Stephanie?
Hi, good morning, everyone, and thank you so much for that introduction. So before I begin, I just wanted to show my FDA standard disclaimer slide here. Next slide, please. Okay, so to begin with, for my part of the presentation, what I'm going to be covering is really some background information on why cross-study analysis is important. I hope to go through a variety of different case examples to give you an idea of how cross-study could be used. And then I'm going to touch upon how a tool can be designed to provide the overall ranking or scoring of toxicity profiles across studies, across drug classes, and across applications. And then I'm also going to touch upon what we were thinking about within this collaboration between BioCelerate and FDA in terms of how we define this process. How did we go about with this proof of concept for this cross-study analysis tool and app? Next slide, please. So before we talk about cross-study analysis, let's just kind of back up and ask, in general, how is non-clinical toxicity assessed? Because that was really our starting point with trying to design this particular app. So when you consider how an individual, an analyst, a toxicologist, a scientist, would look at study data, what are the processes that we are going through? So of course, the user would look at that study report. That study report would have a certain study design, certain species, a certain drug, and then there's a whole lot of data to get through. And again, this is a manual effort. So there's everything from looking at body weights, clinical signs, mortality, to all of the numerical data, such as looking at clinical chemistry parameters, looking at histopathology, and putting all of that information together to try to come up with, well, what do I see here in terms of a toxicity profile? So the user, the end user, what we are doing with looking at throughout this entire process, throughout this entire study report, is to ultimately come to a conclusion, state that all right, the target organs of toxicity are X, Y, and Z, and the severity of these toxicities have some type of ranking in our own mind. If we're comparing that to, let's say, another study or another compound or whatever we might be analyzing. Next slide, please. Okay, so thinking more about this, how is non-clinical toxicity assessed? Well, it's rather complicated, and we do a lot of different calculations in our own mind, and we do our own assessment. So, for example, if you're looking at the kidney, perhaps you're looking at multiple different parameters. Let's say you start with something like clinical chemistry, and you're looking at clinical chemistry values. So you're looking between control across dose groups, maybe you're looking at high dose versus control. But the process that one goes through is to really assess, do I see a difference here? Is there an increasing trend, a decreasing trend? What's really going on here? But this is numerical. You might ask, is this statistically significant? And then even if it is it biologically meaningful? And that's based upon your own particular experience. So let's say you note something there in clinical chemistry that might be something there. Well, maybe you move on and you start looking at the actual organ. So you start looking at the kidney and the histopathology findings. So how does one assess that? Well, again, it's a little bit complicated. Perhaps you're looking at kidney histopathology findings, and your first question may be, well, am I going to assess, severity of toxicity to, necrosis? Is that more important than maybe mineralization? And so maybe that's driven by the incidence of that finding, and it's maybe also driven by the severity of that finding. So you're not only accounting for incidence, but also severity. You may also be looking at, well, what species is this? What sex is this? Because based upon your own experience, there may be explanations for some of these findings. So again, it's a little bit complicated in terms of how the user, the toxicologist, may be looking at the non-clinical study information. Next slide, please. Okay, so with that said, I hope I've given you the idea of, manually going through this process of assessing non-clinical toxicity. Thinking about applying that to cross-study analysis, well, this can get rather complicated. So you're going study by study by study. Maybe you are, noting down the target organs, what is of particular interest. Maybe you're writing yourself, some notes or creating an Excel spreadsheet. But again, this historically has been a manual exercise that takes a long time to do. So what's new and different? Where are we today? Well, fortunately, we have the implementation of the SEND model. And that really represents a tremendous opportunity for us to apply large-scale data analytic approaches to the non-clinical toxicology studies, to be able to think about designing tools like this. So the idea for cross-study analysis and this proof of concept is really just to enhance the interpretation of the data. So of course, one is still going to look at all of the report findings, but think about if you're looking across a number of different studies and you want to have a better idea about what the target organs are, or maybe what's different between different compounds. This is a way to kind of show you the way. Where is their interest? Where should you dive deeper into the data? So it was really this concept and this idea that a collaboration was formed between BioCelerate and FDA so that we were able to develop these algorithms and data transformation strategies to enable the comparison of toxicologic findings of two or more studies. And in conjunction, BioCelerate, FDA, CEDAR, and Fuse have developed open source search scripts to enable various cross-study analysis use cases. Next slide, please. Okay, so thinking about what are we doing? We're trying to take the manual process and we're trying to apply that logic into a tool, into an app. So again, trying to take the user experience of asking, well, what's that target organ of toxicity or toxicities in a particular study? What logic am I applying to all of these different parameters? And then, applying that into a system where, now we have an automated approach to kind of point us in the right direction. So how does one do that? Next slide, please. Okay, so on this slide, before I kind of jump into the use cases, get you thinking about different ways to use cross-study analysis, I first wanted to go over some kind of big picture ideas about how cross-study analysis could be used. So one of the more common ways that cross-study can be used is certainly understanding toxicity profiles across studies. Perhaps one of the more common themes may be to look across time for one compound over time to see if there's similar or different toxicity profiles across time and across species. But beyond that, you could use cross-study analysis for other things, such as identifying off-target toxicity or looking for trends in a particular pharmacologic class. So both of those things, what is unique and what may be different. But then beyond thinking about a particular compound or a drug, well, what about vehicles? I mean, you could apply the same cross-study analysis to these areas as well. And then also thinking about, could this be used to really understand the frequency of a rare or incidental findings? So if you are, comparing across a wide variety of different studies and you're seeing outliers, you're seeing, something that's happening infrequently, well, maybe that shows you it's there, and then you have follow-up questions, and there's further studies that you do or further inquiries that you do. And then also thinking to the future, thinking about where this could be leading. So the way that I would think about this is, well, what do you learn from cross-study analysis? What additional questions might you want to ask? So thinking about what you learn from cross-study analysis, maybe that leads into future, predicting or modeling toxicity profiles of particular compounds. Maybe through this analysis, you've identified certain chemicals or chemical structures that are responsible for off-target activity, so you could better design something in the future. And then also really important to think about, if we're able to do this with non-clinical data and trying to look across studies and seeing what is similar and what is different, then what about applying that to clinical data? So adverse events. So are there similarities? Is there any predictivity? Is anything lining up? So again, things to think about, for the future with how to apply cross-study analysis. Next slide, please. All right, so over the next several slides, what I'm going to do is kind of walk through different use cases. So just to get you thinking about how cross-study analysis could be applied. And this really can be applied to any question that you have across different studies that you have available to you. So for example, let's say you have one particular compound and you have multiple different studies, let's say 6 different studies across time. You have everything from your 1-month toxicology studies to your chronic toxicology studies, and you have that in 2 species. So you want to apply cross-study analysis, and maybe you simply want to know or see, are there similarities across these studies? Does some toxicity go away? Does some toxicity show up in your chronic tox studies that wasn't there in your acute dose studies? Or are there similarities across your species? So in this example, hopefully cross-study could help to point out that, oh, okay, cardiovascular seems to be a common target organ with this particular compound, and that carries through over time. Whereas maybe later on, maybe 6-month tox study in a rodent, you see some new CNS toxicity that you hadn't seen before. So again, it's just a way to try to point out similarities and differences over time of a particular compound. Next slide, please. So in this next example, let's not talk about the same compound. Let's talk about 6 different compounds, but maybe they are all in the same pharmacologic class, and you have the same question. Maybe you're looking at 6 different compounds, they share the same class, but you're really looking at the rats. You want to look at 6 different rat studies, all from the 1-month tox studies, and see, all right, well, are there similarities or differences in the toxicity profiles across these studies? So again, in this hypothetical example, you look at your 6 studies, and once again, you see that there's cardiovascular toxicity in pretty much all of these studies. Well, that may tell you something, but maybe compound number 5 is really interesting now because, well, it doesn't seem to have that cardiovascular toxicity finding. So maybe you want to dive deeper into why that is. So again, pointing out the highlights of what is the same and what is different. Next slide, please. Okay, so now let's make things a little bit more complicated. So now let's say you have those 6, same compounds that you're looking at, maybe all the same pharmacologic class, and now you've added in 6 more studies. So now you have all of your non-rodent studies, maybe different species. So now you're looking at, let's say, 3 different species, but all 1-month tox studies. Maybe your question is, okay, well, I saw this cardiovascular signal in the rat. Does that extend out to the non-rodent? So now with cross-study analysis, putting all of these, on the same playing field, so to speak, could you be able to see that, oh, all right, well, that trend is continuing across different species with the cardiovascular findings? So again, what is the same? What are the signals that I'm seeing as these number of studies, continue to grow? Next slide, please. So to continue on that theme, same data set, maybe your question is, I want to know what's different here. So aside from the cardiovascular system toxicity, Maybe what you're seeing here for the first time is you're identifying that, okay, well, compound number 6 is a little bit different. There's this new different CNS toxicity that we're seeing. Well, maybe again, that leads to more questions. Why is that? What is, the chemical structure? What's going on here? Is this off target? And then also, maybe you're observing that in your dog studies, your 1 month dog studies, now you're seeing GI toxicity, where you're not seeing that, let's say, in the monkey. And maybe that is, an expected finding for this drug class. And then, maybe with compound 1, you're seeing in the monkey, there's some new respiratory finding, and that's different from everything else. So maybe that leads to additional questions that you may have. So again, it's really adding more studies, seeing what's similar, seeing what's different. Whatever question you have, you can apply it to this system. Next slide, please. Okay, so in my last 2 slides, I just wanted to make things, really complicated, and, where can this be taken? What are the extremes of this in theory of how this could be used? So again, let's say you have now your 6 different compounds, all the same pharmacologic class, more time has elapsed and you're revisiting all of this. So now you're not only looking at your 1 month tox studies, but you have different compounds, you have different studies completed. You want to see what's really happening now in your longer term studies, in your 3 month studies, in your chronic tox studies. So how do you find the signals in the noise? How do you find any similarities without doing this as a completely manual effort? Next slide, please. So again, my last slide in this series is really just to showcase that, again, the intent of this cross-study analysis is really just to point the way. So now you have a whole lot of different studies, let's say more than 20 studies. How are you going to get this under control with all of these data sets? So again, the idea is to feed the information into this tool, into this app, and then the output is simply to show you where things are aligning or not aligning, and then lets you, the user, follow up on any of that. Next slide, please. Okay, so with this slide, I was just trying to, summarize everything that I've just said pretty much. So the concept, the cross-study analysis concept, is that you can compare whatever you want to compare. Ask whatever question you want to. You have a variety of different compounds, a variety of different studies. You can look at the same compound over time across species. You can detect differences and similarities by study duration, by sex, by species, by strain, or by particular compound. And the end result is really to give you information on the target organs of toxicity, as well as the severity of those findings. And that is really the intent. Next slide, please. All right, so what are the key pieces that we need to be able to do this? So the first is we need a source of data, of standardized study data. So again, SEND allows us that opportunity. So SEND, the Standard for Exchange of Nonclinical Data, has really been the key in being able to do these type of projects and look at these analyses. The next question is, well, how do you rank or score your findings across the study? How do you compare a liver toxicity in a rat to liver toxicity in another rat or another dog, or whatever you're comparing? So the answer here is you need to establish a scoring system. And importantly, you need to establish a scoring system that is user modifiable. So the user has the experience. The user should be able to input this information. And then finally, how do you select the studies and the criteria and view the results? So from a practical standpoint, what do you see as the user? Well, we needed to think of a way to create a user interface or a dashboard. Next slide, please. So with that said, and thinking about user input, what does the user need to consider? So the first is the source of the data. So the user should be able to determine what studies you're going to look at. What dose groups are you going to compare? What sexes are you going to compare for your analysis? So again, allowing you, the user, to be able to change those parameters. And then also data selection. So maybe you just have an interest in the liver and the kidney, and that's what you want to put into the system. I just want to look at these 2 organs. You want to look at specific endpoints. So you, the user, should be able to decide, what are you trying to identify? What do you want to see? And then finally, the scoring methodology. So again, an interface that will allow you, the user, to establish a scoring system for endpoints in each send domain to create these visualizations that should help you in your analysis. Next slide, please. So with that said, what I want to do with this slide is really just to introduce you into the concept that you will be hearing much more about in this presentation. And that's, what does this visualization look like? What did we come up with? So as I just stated, the user will input, certain defined requirements and information into the system. And the output, there are multiple outputs. And the first is a very high level overview or high level visual. And what we came up with was a radar plot. And so basically, I just want to introduce you into what you're seeing here. So with these radar plots, you have the different organs of interest that you have selected. So in this hypothetical view, you're looking at liver, kidney, brain, GI tract, and lungs. You're looking at 3 hypothetical test articles. Let's say these are all 3-month female rat studies. And so the different colored lines are indicating, the different severity of the target organs. So for example, if you have test article number 1 and test article number 2, you'll see that these are kind of, clustered around the center of this radar plot. Well, that really indicates for all of these different target organs that there's really low incidence and low severity of findings in really all of these organs, not much different from control. But now you look at test article number 3, and that one is, has much different profile. You see that, the lines are extending out further away from the center, especially for the liver. And what that indicates is the further away you get from the center, you have an increasing score. And as you have an increasing score, that indicates you have an increasing toxicity in that particular organ or organ system. So how I would read this is that test article number 3 has a significant toxicity profile in the liver, followed by the kidney and the lungs, GI tract, but then the brain, CNS, not so much, pretty much similar to the other 2 test articles. So that's the concept. These visualizations kind of lead you in the right path of what's the same, what's different. And then from there, being able to drill down into more information. So looking by send domain, so for the liver, for example, what's feeding into that scoring, and then by individual study parameter. Next slide, please. So this is my last slide, and I just want to walk through this for you, just to keep in mind about, during this presentation, this is the concept that we created. So again, thinking about looking across a number of studies. What do you want to see? You want to see similarities, and you want to see differences, and you want to see how that compares across different organs or organ systems. So that first tier radar plot is really just supposed to direct you to say, all right, well, maybe all of these products have a significant liver toxicity. Maybe I want to understand more about what's driving that score. So now there's the ability to drill down. Now you get this second tier radar plot. So now if we take the example of the liver, well, what's feeding into that score? Well, from SEND, it's the scoring from clinical chemistry, or the LB domain. It's the score from the histopathology findings, or the MI domain. And it's the score from the organ weights, or the OM domain. So beyond that, let's say you want to know a little bit more. So for clinical chemistry, what's really being calculated for that? Well, that's being calculated using values for ALT, AST, ALP, and albumin. And maybe I, the user, want to see exactly what those values are. So that allows for individual study data to be visualized as well. So the overall concept is that you are taking multiple different studies, asking whatever question it is that you want. And then you have this path forward to be able to say, first of all, what's the same, what's different, and then drill down into the information from there, which can, lead to answers or can lead to more questions for you. So with that, I would now like to turn this over to Dr. Bill Hauser to continue the presentation.
Thank you, Stephanie. That was thanks for taking us through such an important conceptual basis for what we've put together here. um, as she went through, we went from this top tier radar plots all the way down through the individual study data. What I'd like to um, talk through is going from the individual data, how we went from there to in our this tool that we have created to summarize it as we go up. So as you can see at the bottom, we have this individual data. And um, the let's go on to the next slide. The data that we actually look at is not very well standardized or normalized in the sense that there are wide ranges of values for various types of measurements. As shown in the example here for liver enzymes, there's a much wider range of that are considered normal compared to the ranges for the electrolytes such as sodium and potassium. And what we're in these uh, radar plots, we want to be able to show the what is a significant variation from acceptable values, like normal, no toxic effects, versus ones that are more substantial. And so the next slide describes how we found a way of dealing with that. So on this slide, we see the z-score normalization described. Z-score was is a method that we used to get these all similar. On the left-hand side, we see an equation that explains how we compute the z-score value for an individual measurement. Um, what we do is we take that measurement, x sub si, which is a measurement on a particular study for an individual animal at a particular point in time, and we subtract from that the average measurement of the control group for that same study, divided by the standard deviation for the control group. So with that subtraction, it takes the variation in the numerator there, the measurement minus the average, moves everything to the same baseline effectively. For all the measurements, it's showing the difference from the control's average. Then dividing it by the standard deviation of the control changes the, the magnitude of the variation. So in effect, we are converting the individual measurements from the variations that they have into being the number of standard deviations away from the control's mean. So this center portion of this slide describes what we would expect for a distribution of the z-scores within a study for uh, if there was no treatment effect, assuming that the measurement had a Gaussian distribution. So it is exactly the Gaussian distribution. If there were a treatment effect, we would expect the distribution of the z-scores for a treatment level to be shifted to the right or to the left, or maybe broadened or narrowed. So as a theoretical example of how to compute a z-score, let's consider an individual dog that may have a serum ALP value of 50 units per liter. And the control group on that study had an ALP of 70 with a standard deviation of 40. So for that dog's ALP value, we would take the 50 minus the average from the control group and get a negative 20, and divide that by the standard deviation of the control group, which is 40, giving us a negative half. Since this value is half of a standard deviation lower than the control group mean. The next slide, please. So here we show those z-score values for a number of the liver enzymes and related measurements. So we show them, and this is an example from the tool. In the gray area in the center, we've grayed it to show that this is very close to the control group values, and so doesn't really show a significant variation from the control group. On those points that are way far to the right at 4, a z-score of 4, those are very significant variations from the control group, and -2 is a notable change. So this doesn't tell us the actual values, but it does give us a very quick example way of visualizing what is a noticeable change and what is a not much of a change, which is very helpful in as we create the higher level visualizations. The next slide, please. So that's the beginning of our normalization process. Then here we describe the more additional normalization that is needed. For most of the numeric values and all the measurements, we end up creating what we call a TOX score. And the TOX score for numerical measurements is closely based on the z-score. So the z-score that we talked about on the previous slide, we ignore the plus or minus sign by taking the absolute value of it. And then we ignore the fractional part by just looking at the integer portion. And that gives us the score, the z-score. For body weights, we need additional standardization because we consider that what's actually more important than the individual body weight is the change in the body weight from the last day before treatment up through the base, or the last day of treatment minus the baseline as being the change. And so we use that as the X values going into the z-score. For organ weights, similarly, we consider the organ weight divided by the brain weight to be a better indication of the organ weight impact than the individual values. The microscopic findings present some additional challenges because, of course, we have the severity of the scores, of the findings, as well as the incidence of the findings. And so we used the maximum of either the individual of the incidence for that group, the score from that, or the individual's severity score, because we didn't want to have an individual's significant value to be kind of washed out with an average across the group. So the individual severity scores are used to determine a TOX score, and the group incidence was used to compute a TOX score, and then the maximum of those was used for that individual's TOX score. The next slide, please. So with these values, we could then create like a liver radar plot. We show the summary of all the lab values on one spoke, the organ measurements on another spoke, the microscopic findings on another one, and we decided to throw in body weights as an additional measurement for context. And so the next slide, please. Then using similar radar plots from the liver and from the kidney and body, the reproductive system, we can consolidate those spokes into this higher level scoring system. And there are 3 ways that we enable them to be combined together. One is by using the animal max values from the previous plots to be averaged, to be combined together to make the spoke for here. or using the endpoint maximum, regardless of whether it's from the same animal or not, or just the average. So as again, like Stephanie was pointing out, any value that's at the near the center is 0, a change from control. And as it moves out, it becomes more and more of a variation, whether it's a variation to the right side or to the left side, a positive or a negative. Either way, it becomes a larger variation from control and becomes from not significantly different from controls to being more significant enough to being a marked difference. So from here, I think this is my last slide. I'll turn it over to Mark to describe in more detail how we use such a system for interpretation.
Thanks, Bill. Yeah, so I will take over from here and talk about the actual results of our analysis, at least the way we set it up, and sort of walk you through how one might work through the data and make some interpretations of the data. So if you could go ahead 2 slides. One more, please. What I'm going to do is tell you a little bit about the data that we actually used um, for the analysis. Could we go ahead one more slide, please? Yeah. So the source of the data that we used for our analysis is send formatted data that was provided by BioCelerate member companies. Uh, BioCelerate has a data sharing consortium with member companies where we share toxicology data, and we found in that data set that we had 2 different compounds intended for the same pharmacological target. Um, and those studies were rat and dogs, 1-month rat and dog studies um, for that particular pharmacological target. In these slides it will show, and we've been showing, they're designated as compound A and compound B. What we did for just sort of this proof of concept analysis is that we looked at high dose male animals, but I'll show you as we work through this that the user has many options on how they may want to visualize the data. And then finally we went back and had uh, toxicologists review the study data for the 4 studies that we analyzed and confirmed that the interpretation of the toxicology studies on an individual basis was consistent with the output of the tool and the findings that were identified from using the tool. Next slide, please. So the way we set this up, this is in our Shiny app that we set with many options to make different selections of data, organ systems, scoring, how you wanted to score it. And we made this all so that it could be modified. We set up these cross-study analysis organ systems, kidney, liver, hematopoietic. Again, the list of tissues and endpoints that can be used for them are modifiable by the user. The intent here is to create a tool that can be used by scientists in the field for to do judgment-based interpretation of the study. The tool itself doesn't interpret, is not it does not provide an output, for example, of it. Oh, this is a compound related adverse effect. I mean, it's not designed to do that. It is designed to enable the user to work through the data, to expose patterns or consistencies or lack of consistency between studies and data points, for the expert to evaluate and dig into further to understand and enhance the interpretation of the study. Next slide, please. So the dashboard we set up, this our Shiny dashboard, it is set up like this. And right now we have the 4 studies, so the 1-month studies in rat and dog for compound A and B. One could select those. As I indicated, we for this proof of concept, we focused on high dose and high dose males. And then we built a number of organ systems, and again, it's all transparent in the tool as to what we included for example, liver and kidney and hematopoietic. So we have sort of check boxes for those to pick which analysis you would like to do, and then also the options for the scoring and modifying the scoring patterns. So fairly straightforward as far as selecting data. Obviously, a little more knowledge of the tool is helpful when you're getting into modifying scoring and ranges and things like that. But you know, user friendly, our Shiny app. If you go to the next slide, please. So this is the initial output. Bill's shown this. I think we've shown this a couple times now, but now I want to take a look at it from a toxicologist or a scientist that is interested in trying to understand relationships and differences between these 2 compounds. So this radar plot, you can see the spokes. We have liver, kidney, body weight, reproductive system, endocrine system, and hematopoietic system. So this is our first really high level look at the data, trying to get an impression of, obviously as a toxicologist, we think about, are there things that could be on target or off target, right? On target things in a radar plot like this, at least at an organ system level, would be, dots that are all significantly moved from center and grouping together. Um, Kidney sort of hints at that. We have to dig into that more to see if there really be something that would consider on target for kidney. Then the other side of it is one compound potentially have an off target toxicity or a different toxicity that we need to look into further. And you can see here the compound A, particularly in the dog and rat, seems to sort of separate in the radar plot as it's further out in the spokes, indicating that there are bigger changes with those compounds. And you can see particularly with liver in the dog that there's some things there that are driving that Z-score higher. We'll dig into that in a minute. And then you can see on body weight, both the rat and the dog had, a much different effect, much more of an effect on body weight than the compound B, dog and rat. So, there's many ways you can look through this and start to pull it apart. What I'm going to do for this example is we're going to go and follow liver and see what the sort of the basis, or get some ideas about why compound A seems to be separating somewhat from compound B with regard to liver. So the next slide, please. So this is a radar plot we created just for liver. So the points here are OM, which is organ weights, body weight, MI is the microscopic evaluation, and LB is the clinical chemistry evaluation. And again, we start to see some of the details of why the compound A dog data is separating. We see that there appears to be some MI findings that are driving that higher Z-score. We also see that there are also some clinical chemistry findings that it appears that are driving that, and it confirms the body weight finding we saw in the high level plot that again there appears to be some differences there that we should look into. And it also showing that compound B, it appears that the dog organ weights seem to be tracking with some changes in the compound A dog organ weight. Um, so we'll dig through these one by one. So the next, go to the next slide. Um, so this is body weight. As scientists that interpret data from toxicology studies, we often want to see body weight as one of the first things that for our initial evaluation. And as we all know, when you look at body weights between rat and dog, you can't plot them on the same plot. It's hard to compare them. However, here with using the z-scores and doing a baseline subtracted body weight, we're able to plot dog and rat on the same plots and be able to evaluate all four body weight plots together. Um, and you can see here that what we saw in that initial radar plot, that the body weights for compound A were apparently different than compound B. That's showing in these plots where both the dog and the rat body weights seem to decrease fairly significantly over the time course of this 28-day study. Um, the plot on the bottom, is more, it's in a format where we're looking at percent of change. So that sometimes is more easily interpreted by toxicologists and scientists familiar with toxicology data. So there's two ways to do it. We did it with z-score and percent change. Again, both confirming what we saw in the radar plot and providing us some additional detail as to, how quickly did the body weight decrease, and what was the extent of it. So that was our first evaluation, is that these compounds are clearly different based on their effect on body weight. Next slide, please. So this is a look at the ClinChem z-scores. So the way this is set up, and Bill showed a glimpse of this also. So z-score 0 in the middle is no different. And the solid lines up and down are the z-score mean for the four data sets. So you can see, as like at the top at ALT, the compound A dog had a fairly high z-score for ALT. So it's pulling that mean for that group to the right, outside of the z-score normal range. But then also we can see sort of the rank order of changes in that, both of the species for compound A saw, or at least the dog, there was a high change in ALT. The others were probably within standard limits. And again, that continued for ALP and AST for the dog study. So the changes in ClinChem for that dog study for compound A were really driving the changes that we saw in the radar plots. And again, we stopped at this level as far as the analysis, but at this point you would want to dig further and see how high those ALT levels were, and ALP and AST, and really understand what exactly was going on with those ClinChem values. But again, it's allowing us to visualize and look on one plot, look at the four studies, and understand potential differences and similarities. If you go to the next slide, please. So this is the organ weights. This is brain ratio weight z-score. And again, this is playing out what we saw in the radar plot, the liver specific radar plot, where compound A and compound B dog studies had significant increases in liver weight. And that's what's showing here in this z-score plot. It appears that in the rat there really wasn't any significant differences in liver organ weight. And again, as an interpretive type part of it, indicating that there's some kind of species related effect here, that for this target that we're seeing organ weight, liver organ weight changes. The next plot is I believe is the microscopic evaluation. And what we did for microscopic is, next slide, please. What we did for microscopic was, um, tried to get incidence to portray incidence and severity on the same plot, which is difficult whenever you're looking at tabular data from a study and trying to understand how to compare both incidence and severity across multiple studies. So we come up with this plot. On the bottom, we have the microscopic terms that were identified. This is liver. For this case, it's liver. So you can see on the bottom all the various findings that were called by the pathologist, and then on the left was the various compounds and vehicle groups. And when we look at this, remember we saw in the radar plot that um, there were some microscopic findings that were driving a higher Z-score for the dog compound A. And here we see that pigmented macrophages were, occurred at a fairly high incidence with a sort of a moderate severity. So that could certainly be driving some of that score. There was also accumulation was identified, sort of a moderate incidence, moderate severity. So again, these are things that one would want to dig into deeper and look at perhaps individual animals or other findings in the other species to try to understand sort of the origin of these findings. But again, these findings are driving those R scores to enable investigator to assess the potential impact on interpretation of the studies. Next slide. So sort of wrap this up. We've used this slide a couple times to try to give the framework for how we work through these. Um, we focused on the liver toxicity profile here. As I said, we have profiles set up already, and the user can change those profiles and create different profiles to do their assessment. But the power here is that being able to compare across multiple um, organ systems and endpoints to try to interpret these studies. As Stephanie pointed out in the beginning, um, up until now, up until the availability of SEND data, this has been pretty much a manual process that individual scientists work through to try to interpret studies, to inform dose selection for future studies or interpretation of on-target versus off-target effects. Um, and we feel like this tool is a step forward to enable a more thorough and deeper analysis of the data to enable that judgment-based interpretation of the data. So next slide. To summarize briefly what we've been through. As we emphasized, we've created this R Shiny app and this analysis platform with the user-defined thresholds. This ranking of severity and sort of being able to compare across species has enabled the ability to look at multiple studies at the same time without being being lost in the magnitude change, the differences in parameters between species. As we look through this, at least looking at the output from the radar plots, it appears in some cases, as a potential interpretation is that there's liver, kidney, and hematopoietic system effects that may be on target, but then there was also some effects that appeared off target. And again, it relies on the scientists digging into the data and making those judgments. But we really think that this is an improvement, as far as understanding what's related to the target, on target, off target, The use cases we talked about earlier about duration of dosing. Do we have progression of findings? This tool could be used to understand those types of things. Species differences. We showed species differences here. So again, the goal was, we have the access to send data now, this digital data, and a lot of the early emphasis on send data was to interpret individual studies and analyze individual studies. And we felt like there was an opportunity also to extend that analysis and evaluation to multiple studies. So with that, we showed the use cases earlier, and Stephanie alluded to, our ultimate goal as scientists is to understand the translation of non-clinical studies to clinical studies. And if you go to the next slide, so based partly on our results of this evaluation and other things going on in the industry, BioCelerate has kicked off a cross-functional clinical, non-clinical project to enable translational safety and translational evaluation. And part of that initiative is to incorporate tools like this, the cross-study analysis tool. We feel like that we are getting to the point with digital data, both clinically and non-clinically, that we can apply tools like this to sort through multiple studies, uh, provide information on similarities and differences, and you can get a better understanding of what really translates from the non-clinical studies to clinical. So again, this is a large initiative that um, BioCelerate and TransCelerate have kicked off in the past 6 to 8 months, and we look forward to uh, letting you know in the future, the progress we're making on this and some of the use cases for translational safety. All right, next slide. And this is just to provide you, we're going to provide the slides to everybody that participated. And these are some links to the manuscript that was recently published on cross-study analysis. There was also a manuscript on the our Shiny app. We call it Send Digger is one of the tools that we use to sort data and analyze data. And then the Shiny app, the historical control collator. So all these links are available and for public use and available for individual scientists to use as they wish. And I think with that, we're ready for questions and comments.
All right, thank you, Mark. If I could have our panelists join me on camera, and we'll dive into some Q&A here. As a reminder for the audience, um, please submit your questions up at the top right-hand corner, and we will. And if you could also indicate who the question is directed towards, we can route that accordingly. First, I'd like to start off by thanking our panelists, Stephanie, Mark, and Bill, for a great, engaging presentation and for helping demonstrate how we're trying to advance the use of SEND standards in this manner. So the first question I have is for Stephanie. How do you see a tool like this helping the most with the work you and your colleagues do?
So yeah, that's an interesting question. So from my personal perspective, being at FDA, we have a lot of data, right? So, and there are a lot of questions that arise. So having the ability from a very high level to be able to look across studies in the same pharmacologic class, in the same program over time, just to better understand what's happening, so that we can we can use that information to inform our policy and processes and internal engagement with our reviewers. So again, it's this concept of cross study, of really being able to dig down into a lot of different studies to see patterns, to see differences, that I think will help inform everyone and will benefit everyone. So from my perspective, I mean, of course, reviewers could use this type of tool, absolutely. But I think of this as a broader perspective. So being able to look, across large data sets, and answer a lot of different questions that we may have.
Thank you, Stephanie. Yeah, it sounds like an efficiency and quality gain all wrapped into one. So that's great. Um, Next one, I'm going to ask Mark to jump in here. So great tool. The demo version of the app we tested was hard-coded to several studies shown in the presentation. Will there be a version distributed that is easily adoptable with new or other data?
Right. Yeah, I'll take a shot at this and maybe ask Bill to fill in also. But yeah, the R Shiny tool is designed to um, is to intake any type of send data. So I think the user can provide their send data sets and link them to the R Shiny app and do analysis with their studies. We simply hard coded the the four examples in our case to for the proof of concept.
Mhm. Yes, we certainly welcome collaboration in the development of the open source tool. So uh, you're welcome to extend it. But yes, we have visions of enabling this tool to be used more broadly.
Great. Thank you, Mark and Bill. The next one I have is for you, Bill. Is there any connection between the SendDigger package that BioCelerate FDA Infuse created and this tool?
Yes. Um, no, not at the current time. It's as the previous question alluded to, it is separate. Um, Certainly with SendDigger, it has the ability to easily bring in Send data sets. And we also have a vision of making these two ways of looking at the data more combined into a single tool. It's a vision at this point, and we again encourage people that would like to help that to happen to join us.
Okay, thank you, Bill. Um, Next one, back to Mark. Can we utilize the send data for target identification?
Um, I'm I'm assuming that question is on target toxicity. I'm not totally sure what is meant by target identification. Um, but I could see as one could use, if you had multiple data sets um, from different targets, um, I think there's a potential to, at least with the radar plots and some of the analyses, to see if there's groupings of findings based on the various targets that may be included in the data sets that you're using. Um, so I think again, yeah, using the sorting and grouping, one could create outputs that may guide them into understanding certain target effects or off-target effects.
Thank you, Mark.
Mm-hmm.
Okay, with that, we have concluded our questions. Um, oh, wait, never mind. I had a couple more come in, so we'll just take a chance to review those in a moment. And if there are any others, please feel free to send them forward. Um, okay, so first, a compliment. Wonderful work by all the panelists and a brilliant publication. Bill, I'm going to send the question to you. Actually, this looks like a two-parter. So I'll start with the first part. Regarding the limitations of the Z-score, are there thoughts on how the next iteration of the R-Shiny app can incorporate more sophisticated analyses?
Um, so uh, the being an open source tool, it certainly enables any kind of additional analysis that anyone could conceive of. Um, the so it certainly has the ability to be expanded like that. And I guess part of this question is what more sophisticated analyses we might want to have considered. We haven't discussed any specifics as a replacement for Z-score at this point.
Okay, that's fair. And then I'll open the second part to all three of you, if anyone, whoever wants to tackle this one, because I think it's a broader implication. From a broader perspective, do you think these tools can realistically lead to increased transparency regarding tox data across all industries? Would would assume each industry would still try to keep their historical data for each party, FDA, industry sponsor, et cetera, close to the vest. So this is more of a broader transparency question, I think, that I think is appropriate for any of you to answer if you'd like.
Yeah, I think there's an opportunity to be more transparent. At least at a high level. These types of analyses, you can um, you could provide information that could be shared across, in a transparent manner that may not disclose a lot of the details that make people hesitate to share data. Um, so I think there's an opportunity there, um, as far as just using using sort of these high level analysis that sort of blind the people that, the individuals that would see the data from the details that may unmask it from, even like in our case, we could do this without disclosing the target, but still get valuable information from the study. So I think there's an opportunity there for sure.
Yeah, and I would agree with Mark as well. I think there's an opportunity. I mean, certainly each industry, has the opportunity to use this tool, for their own internal purposes. From an FDA perspective, I mean, of course, if we were to run some type of analysis, it's all anonymized, there's an end result, those types of things can show up in publications, in presentations, things of that nature. But yeah, there's an opportunity, um, for that, but again, in a limited capacity.
All right. Looks like we have another one coming in here. Just take a look at that. Okay, well, while we're assessing this one, if there are any others, once again, the top right-hand corner of your browser will be the place for you to submit that. Sorry for the hold here.
So yeah, just to fill the time a little bit here, a tool like this it opens the door to enabling uh, a kind of computer-assisted toxicology. In other areas, we've seen where computer-assisted design has really revolutionized the whole design process of products. In a and here we can see that we can apply some of those same kinds of um, efficiencies to toxicology. So I'm excited to see what this may develop into.
Agreed. Thanks, Bill. Um, okay, this one I'm gonna start with Mark, and again, if others have thoughts, feel free to weigh in. As you know, for POC development, we would need adequate tox study data. Is there an open source database where we can extract data to do so?
Yeah, I mean, there's not. The short answer is there's not an open source database where people provide data to be used for these types of analyses. It was one of the drivers why we worked very hard to make the 4 studies that we use for analysis public, um, and hopefully try to encourage others to make their data public and accessible for these types of analysis. But um, right now there is no public source of toxicology send data that I'm aware of anyway. Um, so again, like we said, the best source for companies that have data obviously is your database, um, but then also being advocates for looking for creative ways to share data so that we can advance the science of this type of cross study analysis and eventually translational sciences. So, but yeah.
Okay, thank you, Mark.
Mhm.
All right, so we still have a little bit of time left if there are any other questions, but without any in the queue, I'll allow for another minute or so before we advance to formally closing. So if you have any last minute questions for the panelists, please do submit those now. Okay. Not seeing any at this time. I think we will move to formally close, and we do have some closing announcements. So you do have a little bit more time to sneak one in if you have one. But again, I want to take the opportunity to thank Stephanie, Bill, and Mark, and thank you not only for the presentation, but all the hard work that's occurred over the last several years, because this has been a years long endeavor. And, I think, to Mark's point, this is foundational work for what BioCelerate is undertaking right now with regards to translational safety. So I think it's exciting from that standpoint that the work hasn't stopped. It's just morphosized into something different at this point. And We have a couple upcoming webinars from the TransCelerate side that we would like to share with you now. So if I could have that next slide there. We have 2 to share with you here. One is in regards to mastering the master protocol and advancing platform trial research. This is a exciting new initiative that has been undertaken by BioCelerate or by TransCelerate, I'm sorry. We have a esteemed group of colleagues there that will be discussing that next Tuesday. So feel free to scan the QR code there if you're interested in registering for that. The second one on the next slide is Digital Data Flow Solutions Showcase, and that is coming up in a couple of weeks on September 26th. Again, this is a fairly newer initiative on the TransCelerate side, looking to digitize the preparation of and execution of clinical research. so that we can try to remove manual processes and make it more efficient. So again, some exciting colleagues there that will be joining us for that conversation as well. Once again, in partnership with CDISC there, and again, QR code for you to scan if you're interested in registering for that as well. So with that, we will thank you once again for those that joined today. Again, referencing the document with the links up at the top right hand corner of the screen, the materials section, and if you have any questions that can get submitted before we close, if we don't get to them, we will certainly follow up with those as well. But thank you for your time, your attention, and your participation today. We really appreciate it. And on behalf of the panelists, we wish you a good day or evening wherever you are in the world. Thank you so much.